Hello everyone, my name's Steven, and this is an ASTS how-to video on how to study for genetics, specifically on the concepts of mitosis, meiosis, and cross-linkage. So what is mitosis? Mitosis is the process by which a cell splits into two new daughter cells, as shown in that figure on the right. And this can either occur in replicating haploid cells or diploid cells. And so the best way to kind of remember this is if you have a haploid cell, it's going to have N amount of genetic material, or if you have a diploid cell, it's going to have 2N. It's going to have two copies of that genetic material. And then in order to replicate and form two new daughter cells, it's going to have to double that amount. So for a haploid cell, it's going to have double the amount of DNA. So it'll go from N to 2N, so that each daughter cell then has N amount of DNA. And then for a diploid cell, it'll go from that 2N up to 4N, so that each daughter cell has 2N amount of DNA. The DNA is always split equally, and it'll make two new daughter cells. A good way to kind of memorize that process is by knowing the phases. And so there are it's changed over the years. There's been 4, there's been 5, but in general, this acronym PPMAT, I think works pretty good to memorize the phases. There's prophase, prometaphase, metaphase, anaphase, and telophase. And you can see the prophase, metaphase, anaphase, and telophase on the right, numbered 1, 2, 3, and 4. And then prometaphase is just this arrow in the middle. And you'll cover the details of each more in class. But a good way to kind of memorize those is that acronym and then the accompanying details after. And then this is always followed up by cytokinesis, and then the daughter cells each enter prophase and then begin growing. Conversely, meiosis is like mitosis, but it's conducted in two separate parts. And the end goal is to always form haploid gamete cells. So a key difference in meiosis from mitosis is that there is an aspect called crossing over, and the gamete cells are always haploid. It doesn't matter if you're a haploid organism, or you're a diploid organism, or even a triploid, tetraploid, it goes on and on. The end goal is to have haploid daughter cells. And so in these phases, there's usually two, where you'll have a normal cellular replication process of prophase 1, prometaphase 1, metaphase 1, anaphase 1, and telophase 1. And then this kind of looks like right here, as you can see, it kind of looks like regular old mitosis, where the genetic material duplicates and then gets split equally amongst two daughter cells. The second step also goes from, a good acronym with this would be up to 4 times with up to 4 times the DNA, so from 2N to 4N, with the first replicative stage, first division stage, and then down to haploid with the second. With the second phase, there is going to be this separation into those haploid stages, and there will also be an aspect of crossing over in prophase 2. And we're going to go over crossing over here in a minute. But in general, a good way to kind of memorize this is the first phase of meiosis, meiosis 1, is going to look a lot like mitosis. And then meiosis 2 is going to take you from two daughter cells down to haploid. As part of meiosis 2, there's also an aspect called crossing over. And it does get a little bit complicated, but in general, it ensures that there's a strong amount of genetic diversity in embryos. There are many combinations that can arise, and it's very random. Though some are more common than others, and then this kind of gets into your Mendelian/non-Mendelian inheritance, which you will learn about in class. And you can actually calculate this amount of crossing over to build a genetic map of linked traits. So you might have covered linked traits, but I can briefly mention them here. And so it's basically when two seemingly unrelated traits can be linked per se if they're close together on a chromosome. So let's say this chromosome right here has alleles for a mouse with yellow hair and smaller snouts, but the other chromatid has it with bigger snouts. So then those traits would be linked because they're on the same chromosome. Now this diagram is actually wrong in the way that it's oriented. This chromatid right here should be facing this one. And so basically in crossing over, what happens is you have those chromatids that cross over facing each other, more so like this down here. This one is wrong. Down here is a little bit better. This little bit of genetic material came off of this homologous chromosome during pro-metaphase 1, or sorry, 2, pro-metaphase 2 and meiosis 2. And this bit of genetic material went over to this chromosome, and then this chromosome's genetic material a little bit went over here. This only happens on the chromatids that line up on the inside. And so practicing with spatial examples is one way that I really learned how to do this. Another way to do it is kind of just draw this diagram right here and practice it, recognizing that the chromatids on the outside don't cross over at all, and that the ones on the inside have a strong chance of doing so, but that it's random. And so this leads to a large amount of genetic diversity, and also lets you differentiate meiosis 2 from meiosis 1. So this is a good hallmark of meiosis and the meiotic phases. So from crossing over, we can build genetic maps. And so this is a type of problem that you will see most likely on quizzes, maybe tests, covering genetic mapping and linked traits. And so when you see the word linkage map or linked traits, you might be like, oh my gosh, what is that? What is that? Basically, one linkage map unit, LMU or MU map unit, is just 1% chance of recombination. So if we go back to this chromosome right here, and let's say there's a trait right here and a trait right over here on these chromatids, the chance of them crossing over is determined, genetically speaking, that percent chance of recombination, this physical recombination, the higher the chance of that, the more linked they're going to be, and the higher chance of recombination there's going to be. So for example, if we have a total of 400 recombinations out of, like, let's say 1,000 total offspring, so let's say out of the observed embryos we see, this process right here where this is exchanged for this occurs in 400 of the total offspring, and we can observe it. So then we can figure out the total mapped distance. So we can find the total amount of genetic distance between these two traits. And we do this by just calculating the number of recombinants divided by the total number of offspring. And so we can see that the total chance of recombination between these two traits is 40%. And we can use this to then apply it to a genetic map And we can say those two traits are 40 map units apart. Now, it's important to recognize that this is not physical distance at all. This is genetic distance. And so it's a little bit different, but in general, it confers the probability that there is a 40% chance of these two traits crossing over in any given embryo. And so that's really cool. We can use this to apply it to a whole bunch of other concepts that you'll see later on in the class. But in general, I hope this gives you a brief overview on kind of how to use this formula, what this formula applies to, and then kind of the larger concepts of DNA division. And so you might be asking, okay, so what are some study tips that I can use to kind of memorize this information, categorize it, etc.? So in my experience, I've used flashcards, I've made Quizlets. Both of those have worked to an extent. I think for me personally, the one that's been most helpful is coming up with my own acronyms and running through the pathways. So if we go back to, let's say, mitosis, knowing the acronym PPMAT to go over these 5 phases of mitosis, and then categorizing different things that happen in those phases can really help you understand the process better. You can always also clarify your questions with an ASTS tutor, such as myself, or you can even go to office hours. Office hours are there for the professor to help you if you have any of those questions. And so, yeah, I hope this video was helpful. I hope that this makes your studying a little bit easier. If you have any questions, please feel free to make an appointment with the ASTS tutor who covers genetics and go over this process. Have a good day. Take care. Bye-bye.